lesson today is about the prokaryotic cells. From beginning to the lesson number 9, we study about the eukaryotic cells. And from this part, we discover another cell type. The second cell type in this word is prokaryotic cells. So in this lesson, it's reminding you a little bit about the knowledge from the lesson number 1. So in humans, we are not alone. It means that your body is actually an ecosystem mixing type. And it also means for years or for some definition in the words about you actually consist of the two major types of cells, prokaryotic and eukaryotic cells. And for the human bodies, it's contained estimated 10 power 14 cells. So you are homes of these 100 trillion bacterial cells. 10% in your bodies is bacterial cell, and it occupies 100 trillion bacteria. So the number of cells, bacterial cells, living in our bodies is 10 to 100 times greater than our own bodies. 1 to 3% of the body mass is composed of bacteria. So, for example, if your body is approximately 16 kilograms, it means you're containing 0.6 to 1.8 kilograms of bacteria. And the most cells or the most bacteria living in our bodies they live in the gastrointestinal tract in our digestive system, and there is more than 400 of these microbacteria living in the colons, and it can reach the density of 10 powers 11 organisms per milliliter of the intestinal tract lipids. About the cell size, the bacteria or prokaryotic cells is from 0.1 to 5 micrometer in diameter. So these around here, they have the same size like our organelles like mitochondria or protoplasts. And for the eukaryotic cells, from 10 to 100 micrometer. So why these prokaryotic cells are so small? And you know that the size of some organisms is related to the way they can exchange the nutrition and the waste with their environments. The small nose of free living eukaryote is fecal planktons, is not here. The fecal planktons here. So these organisms so small, prokaryotic cells so small, and if they go bigger, it's very hard for them to exchange the nutrition and waste with their environment, because you know that they are unicellular organisms. Only one cell made up one organism. If they're too big, you cannot exchange enough nutrition and relieve the weight to the environment. I'll remind you a little bit about the classification of the other creature on this planet. We have the 3 domains and 6 kingdoms. We have domain of bacteria, archaeobacteria and eukaryote. Archaea and bacteria, they are made of prokaryotic cells, while eukaryotic, they are made of eukaryotic cells. Okay, so we go first with the first domains, bacteria. Bacteria, they are various diversities in the shape. You can see they can form the root shape, the spherical, the curve, or these basic shape can join to form here two spherical we mix eye, hairs or strings, and they can make a seed, package, or irregular aggregate. They are cells very diversity. They can live in an abundance habitats in the world. They live in the all ecosystem in this planet. So they can live in plants and animals, even the living or the dead plant and animals. They can live in the soil, waters, and in the air. And some types of bacteria can live in the extreme environments like the very hot environment, solid, acidic, or base to base, or the toxic to living. However, the number of bacteria living in the extreme environments is less than the number of the archaea. Okay. And also, they are diversities in the way they metabolize. I have just introduced two ways of living organisms can metabolize. It is the autotroph and heterotroph. Autotroph is producers. The autotrophs are able to make their own food from raw materials and energies. We have autotroph, we have plants, algae. And some type of bacteria, they are also autotroph. And they also, most of them belong to heterotroph. Heterotroph is known the consumer because they can consume the producer or other consumers to make the heterotroph. And there are two different types of the autotroph bacteria. Here is based on the energy source. We have the phototroph, and it's like the plants because they can do the photosynthesis, 可见光合. Or chemotroph is mean they derive energies. So instead of from the sun, they derive the energies from the chemical energies, the chemical reaction to yield the energies. And we can have or classify the bacteria based on the oxygens. Some of them require the oxygen, or we call the aerobics. And some of them requires no non-oxygen, and we call the anaerobics. So anaerobics is very familiar with you, like you do the fermentations, like vegetable or yogurt. So they contain so many bacteria, and they are all non-oxygen based. And also, we can divide the bacteria in the other group, or based on the metabolized nitrogens or sulfurs containing compounds. And next, we go to the structures of the bacteria. So all the cells in this planet, they share four key components. The first three is plasma membrane, cytoplasm, and DNA, and of course, the ribosomes. Ribosome is where the protein synthesis. And for the other components, we have here the cell wall, capsule, and plasmids, here plasmid here. It's about the chromosomes. For the eukaryotic cells, they have the chromosome, and they are wrapped around these protein histones. So remember, we have a 146 nucleotides wrapped around a histone, a protein histone atomic core, right? It's made of 2H2A, 2H2B, 2H3, and 2H4, and together they have a linker histone H1, right? In the lesson about the nucleus, we studied that. Cells, and they mix together, they wrap around, condense so many times they can make a chromosome. It is in eukaryotic cells. But for the prokaryotic cells, they are circular chromosomes. You can see it's like a circle, and the naked DNA means there is no proteins, no histone protein. They are wrappings, surroundings, nothing. So it's more simple. You can see it's very simple. Chromosomes. And because they're very simple, the bacteria can reproduce every 20 minutes. So that's too simple because they replicate 20 minutes. Whereas the eukaryotes, we have a 24 hours. And again, it's because they are too simple, so many mutations can occur during the divisions of the prokaryotes. And I think this 1 hour, 100 bacteria have mutation. So very high rates of mutations, right? Because they have a very simple DNA, very simple chromosomes. They have no protections, right? They have no histones without any histone. Almost time of them is in the forms like the chromatins. It's not like the form of the chromosomes of the eukaryotic cells. And the next structures they both have is the ribosomes. For this part, I give too detailed information. I think it's hard to remember. It's just remember for me, for the ribosomes of the prokaryotic cells, you have a 70S, and it's like 30S plus 50S. And in ribosome of eukaryotic cells, we have the 60S and 40S. Together, they form 80S. That is enough for the questions in the final exams. The next very important part of the prokaryotic cells is the cell wall. Sorry. That's you. We have the cell wall. And there are two types of cell walls of the bacteria. I'm reminding you a little bit, we have, or some kingdoms have the cell walls, we have the fungi, we have plants, we have archaeas, and we have two bacteria. And the cell walls of the bacteria is here, is classified into two types of the cell walls by staining the Gram stains. Stains by Gram staining, you can divide the bacteria into two types: Gram-positive bacteria and Gram-negative. Yes. Other strains by the Gram stains, The cell wall of gram-positive is have the purple colors, whereas the gram-negative have the pink colors. And here you can see that the gram-positive bacteria, they have a plasma membranes, and then they have a cell wall made of peptidoglycan here. Whereas the cell walls of the plant is made of cellulose, the cell wall of the fungi is made of chitins, right? And for the gram-negative bacteria, they have outer layers. The difference is they have an outer layer. And this outer layer makes the pins here. They have one outer layer made of lipopolysaccharides, or we can, the abbreviation of them is LPS. LPS, lipopolysaccharides, is made of lipid and polysaccharides. So the difference between the gram-positive and gram-negative, they're different in colors when we're staining by grams. And second, they have one outer membrane, one more outer membrane. So gram-positive membranes, cell wall made of peptidoglycan. Gram-negative, we have cell membranes, cell wall made of peptidoglycans, and one outer membrane made of lipopolysaccharides. I have a tables to classify the gram-positive and gram-negative bacteria. First, about the cell walls. Gram-positive, they have a single layer made of peptidoglycans, and they're more thicker than the gram-negative. One layer, but thicker. Gram-negative, they are double layer, but they're more thinner compared to gram-positive. Because the peptidoglycan layers of gram-positive is thicker, and sometimes they are multi-layered, so many layers of peptidoglycans. But for gram-negative, they are thinner and often single layers. One layer of peptidoglycans and one layer of outer membrane. So the outer membranes, one absent, one present. Lipopolysaccharide, yes, for sure, because they have no outer membranes. They have no lipopolysaccharides in the gram-positive. Here they have one outer layer made of lipopolysaccharides. So here lipopolysaccharides is present in negative, gram-negative. And because gram-positive have just one layer, they have no outer membrane, so they're more susceptible by the antibiotics. So it's easy to kill the gram-positive compared to gram-negative. Stainings by grams, one is purple colors and one is pink color. Examples of gram-positive, we have a Staphylococcus. Gram-negative, we have these E. coli and Salmonella. So here it is two kinds of bacteria causing the pneumonia. Các bệnh liên quan đến phổi và viêm phổi, gram-positive. And here, E. coli and Salmonella is more related to your digestive system. Các bệnh về tiêu hóa thì thường là E. coli và Salmonella. These ones are, I will show you some candidate. The next structures of the bacteria, we have the capsule. Here you can see the capsule is a smooth layer, smooth surface covering the cell wall. So they're very thick, right? Here the capsule covers the cell wall. And the capsule considers virulent factors because it enhances the ability of the bacteria to cause the disease. Because the bacteria with the capsule can prevent the phagocytosis. So we cannot induct them. It's very difficult to induct and to kill those kinds of bacteria with the capsule. And the second function, the capsule, is will protect the bacteria against the desiccation is in beforehand. So it contains water, it can make the bacteria alive longer, or in these environments lack of water. And another function, they can stick and help the cell attack the surface in its environment. So increase the ability to attack the surface of the target cells. They are very smooth and very hard to destroy by the macrophages. And also it keeps the bacteria live longer. That is three functions of the capsule. And remember for me, this is might be emphasized, they are virulent factors, yếu tố động lực. So the next structures of the bacteria, we have the plasmid. What's different of plasmid to the here nucleoids? They have no nucleus, but have a nucleoid containing genetic materials on the material. However, they have so many small rings of DNA, small compared to the nucleoids. We have the plasmid. The very small rings are, of course, naked DNA. It means that it do not wrap around, it do not protect by any histone proteins. And they are the extra genetic materials of the bacteria. This can pass from one cell to another cell. They can use a vector in genetics engineering. We have the here sort of types of plasmids. We have the R plasmids or resistant plasmid. They are responsible to resist or resistant to the drug. Vi khuẩn kháng thuốc thì sẽ có rất nhiều cái này đúng không ạ? Antibiotic bacteria are here, all plasmids. Next is the sex factor plasmids. The cell have the sex factor plasmid we call the F positive, and they can transfer the plasmids to the F negative. So here we have the donor. We delete the plasmid from F positive to the F negative. The next virulent plasmids. The virulent plasmids is responsible to production of the virulent factors such as the toxins or adherence factors. Next, the heavy metals ion resistant plasmids. They is responsible for resistance from the heavy metal ions, and the bacteria may get exposed in the environment. Next, plasmid for catabolic activities is responsible for degradations of highly complex compounds such as hydrocarbons. So when we put the bacteria in the environment with the high complex compounds, they will activate the plasmid for catabolic activities, and they can digest the higher complex compounds. This is the ways how the plasmids can transfer. The donor cells we call the F plus. They have a plasmid that needs to transfer. Two types of bacteria can make a conjugation. The pili, cái lông của một cái dạng lông của hai tế bào vi khuẩn will make the pellet bridge. And when they contrast, they drawing cells together, makes contact to another. They form a bridge that it can transfer the plasmid from donor to recipient cell. In the second step, one strand of the plasmids was transferred from the donor cell to the recipient cell. And in the last step, the donor will synthesize the second strand, we call the complementary strand, and recipients also synthesize the complement strand to become a egg cells. So in the end, we have two bacteria that contain this kinds of plasmid, and we always call them the egg cells, egg cells. This is how the bacteria with the antibiotics resistance can survive. When they can react to the antibiotics, they can first survive, multiply, and also transfer these antibiotic resistant genes in the plasmids to other bacteria. By conjugation. So here how resistant genes can move between the bacteria because of the plasmids. They can transfer the genes of the resistant antibiotic genes to their neighbors. And the plasmid is the powers, its power tools in recombining DNA technologies. So do you remember the type 2 diabetes? Bệnh tiểu đường loại 2 ạ. Type 2. This is an impairment of the way bodies regulate and use the sugars and the fuels inside the bodies. So if the long-term conditions of the type 2 diabetes, đái tháo đường loại 2 mãn tính, is result into much sugars in the bloodstream and is required to use the anti-diabetic medications, or we use the insulins. We use the insulins to inject into the blood of the type 2 diabetes persons. And the insulins will have the glucose in your bodies in the normal range. So it first isolates insulins from the dog pancreas. So the first isolate from the dogs, and then from the cow, from the cattle, and the pig also. In the past, we need to isolate the insulin from the dog, pigs, from the cow, or other cattle. We use so many years to treat the diabetes. It's saved the millions of lives, but it's not perfect, right? Because it's expensive, and there's so allergy. Uh-oh. And it brings so many allergic reactions. Those are the first genetically engineering synthesis of humans. Insulins were produced in 1978 or 19. 80s, 70s. We can do that. We can do these techniques in 1970s. So the first, we have a genes encodes for the insulins in our bodies. We can take these genes from humans. We have a plasmid. We can open this plasmid. We cut the gene of the human. We put into the plasmid of the bacteria. And it's called recombinant DNA. Next step, we can put these plasmids into a bacteria, and then the bacteria multiply. And the next step, the bacteria, because they have the genes of the human insulins, they can produce the insulin of the human. And then we can isolate the human's insulin from the bacteria. It's more efficient ways. We can make the insulins cheaper and more safe. There's less allergic reaction happens. Cái nào quan trọng lại nói tiếng Việt. Ok, ở đây có plasmid. Ngoài có một cái chức năng của cái plasmid, các bạn tưởng tượng nó là một cái DNA vòng và nhỏ hơn ở trong vi khuẩn, và nó có rất nhiều chức năng. Ví dụ như là ở slide trước thì cô có nói đến là các vi khuẩn mà kháng thuốc, đúng không ạ? Khi nó hình thành thì nó sẽ có các gen kháng thuốc ở plasmid này. Và nó sẽ nhân lên bằng cách là tự nó phân chia cứ 20 phút một lần thì nó sẽ nhân lên thành một khuẩn lạc vi khuẩn mà có thể kháng lại thuốc kháng sinh của mình. Ngoài ra thì con vi khuẩn chứa gen kháng thuốc có thể truyền cho con vi khuẩn không có gen kháng thuốc bằng cách chuyển cái gen qua plasmid, chuyển cái plasmid chứa gen kháng thuốc cho con vi khuẩn khác, đúng không ạ? Cái thứ hai, plasmid này nó có rất nhiều chức năng đặc biệt quan trọng trong công nghệ sinh học, mà ở đây cô ví dụ là trong cái sản xuất DNA tái tổ hợp. Đó là như các bạn biết là căn bệnh đái tháo đường type 2 là mình thiếu insulin, bằng cách nào đó con người thiếu insulin, và insulin thì rất quan trọng trong cái quá trình chuyển hóa đường trong cơ thể, đúng không ạ? Nếu thiếu hụt insulin thì lượng đường trong máu của người bệnh tiểu đường tăng lên cực kỳ cao, và như vậy thì để chữa tạm thời cái căn bệnh đái tháo đường type 2 mình sẽ phải tiêm insulin vào trong cơ thể. Ngày xưa mà insulin lần đầu tiên được tách chiết từ chó, sau đó là lợn, là bò. Cái này nó có thể cứu được hàng triệu người bị bệnh tiểu đường. Tuy nhiên thì nó làm sao? Một là đắt, hai là gây dị ứng, bởi vì nó là insulin từ loài khác. Cái công nghệ tái tổ hợp này ra đời như thế nào? Đầu tiên là trong cơ thể mình có gen để mã hóa cho cái insulin này. Cái thứ hai là mình sẽ lấy plasmid của con vi khuẩn, mình mở plasmid ra, mình lấy gen của người gắn vào trong plasmid của vi khuẩn, tạo thành DNA tái tổ hợp. Và sau đó mình lấy cái cụm DNA tái tổ hợp, lấy cái plasmid này cho vào con vi khuẩn. Con vi khuẩn sẽ nhân lên rất nhiều lần và nó chứa gen mã hóa cho insulin của người, cho nên nó sẽ sản xuất và nó sẽ tiết ra insulin của người. Mình sẽ tách chiết lấy insulin của người và tiêm ngược trở lại vào người bệnh. Cái này thì rẻ hơn, bởi vì vi khuẩn mình có thể nuôi rất dễ dàng, nó có thể nhân lên rất nhiều lần ở trong các bioreactor, và mình có thể tách chiết được insulin của chính cơ thể người. Như vậy thì nó sẽ tránh được các phản ứng dị ứng xảy ra. And the next structure inside bacteria is called the mesosome. The mesosomes aid the foldings of the plasma membrane. They are special membrane structures. They're formed by the expansions of the plasma membrane into the cells of eukaryotic cells, and it helps the cellular formation, adding the cellular respiration. Cell respiration, movement of DNA. And some kind is analogous to the Christie. Remember the Christie of the mitochondria? Christie of the mitochondria, those in the membranes of mitochondria is fold so many times to make the Christie inside the mitochondria. So here, mesosomes is the plasma membranes folding to increase the surface to have the respirations of the bacteria. The next is not really structures, but one process may happen with the bacteria. We call the endospore. Not that the word. The bacteria can form a spore, and how they can do it? When there's starvation, when they have no more nutrition, but they still want alive, until the medium or the environment changes, until they can reach a new source of nutrition, they can next a process we call the sporulation, quá trình tạo bào tử. This is the endospore, how they can form. Here's vegetative growth in normal place. When they have a lot of nutrition, waters, or environment that's suitable for them to grow, we have a vegetative growth. They just divide to make two identical bacteria. Of course, some of them will have limitations, but after 10 minutes, from one bacteria we have two. After more 20 minutes, we have 4. After more 20 minutes, we have 8 bacteria. Is it vegetative growth? But in the case they have no nutrition, or it's in starvation, they are hungry, they will form a process called sporulation. How this happens? In the first step, a septum forms. Septum là vách ngăn here. Septum will form. New one first is not a balance between the two first. Just septum form, new one first. DNA replicates and extend to a axial filaments. And the second step, septum separates 4 spores. 4 spore is mean pre-spore. 4 spore. 4 spore, pre-spore. Tiền vào tự lực. So 4 spore more form mothers. And DNA will come through the septums until they reach, until X compartment get a chromosome. It's divided but not equal. In the next step, mother cells will end up against the 4 spore and surrounding it with a second membrane. Here, the engulfment is here. The mother will end up against the 4 spore. And the next step, the chromosome of the mothers will be disappear, disintegrate. They will destroy DNA of themselves and let the DNAs of the 4 spore survive or maintain the DNA of the 4 spore. The next step, 4 spore develop a cortex layer, tạo nên một lớp vỏ peptidoglycans between the organelles and 4 spore membranes, and the membrane formed from mother cells. The quartz protein deposits outer membrane. The next step, D-picolinate acid synthesizes and calcium incorporates into the 4 spore core. D-picolinate acids and the calcium, here they function to protect DNAs from protect DNA. So they have a very thick cortex. They have a thick the outside is made by Dipicolinix will insert with the DNA and the calcium incorporate in the spore pores. They protect DNA from the heat denaturations and thereby increasing the stability of the DNA. The next step, mother cell relieve the spore, and when they have appropriate environment, they have more nutrition, they stand with the nutrition, they stand with the waters, they can germination again. Nó có thể nảy trồi lần nữa tạo thành vi khuẩn. And this is how explained when you boils the water of your boils your food is 100 degrees Celsius. In some coins you cannot destroy all bacteria because some of them in the form of the spore. It's very stable. It's really difficult to kill the spores of bacteria. And the ways the bacteria can avoid the hot environment, they will keep them alive for a very long time. Example of them, so we can find them in the bacillus or clostridiums or most gram positive. Gram positive can make the anospore. It's easier to kill the gram positive by the bacteria, antibacterials, but in some way it's hard to kill them because when the environments become harder for them, they can form the spore, and it's really difficult to destroy the spore of bacteria. So the next structures of the bacteria, we have fimbriae, we have pili, and we have flagella. So fimbriae, here is fimbriae. So sometimes it looks similar like the pili. I will put the table here to distinguish this one from the other ones. Fimbriae, they are short, thinner, and one bacteria have one from the 200 or 400. But the pili, they are longer, thicker, and they have from 1 to 10 only. Fimbriae is governed by bacterial genes in the nucleolus regions, but the pili is governed by the plasmid. Two types of DNA inside bacteria. We have the DNAs of the main DNAs, we call it in the nucleolus, and we have several types of DNAs inserted in the plasmid. One bacteria have one new DNA in the nucleolus region, but they could have so many plasmid inside their body. Fimbriae is responsive for the cell to surface attachment, enable the cell to adherent surface of the other bacteria. Responsible for bacteria's conjugation is the function of the pili. Here, fimbriae is no receptor for others, but the pili sometimes they serve as a receptor for certain viruses. The virus can hijack this kind of pili and they can invade the bacteria by this pili. So flagellum is just for the movement purpose and it's quite different from two others, so I have no table to distinguish them. Reproduction in bacteria. Asexual. So there is no exchange of genetic materials. We call it asexual. Asexual, they can divide by binary fission. So it is more like the mitosis in the eukaryotic cells. The DNA replicates. They move in the two opposite poles of the cell. Crossword will form here. They form completely and relieve two donor cells, identical to mother cells. Sometimes not identical, 1 hour, 200 bacteria. You have a mutation after subdivision, very high rate, right? The second way of reproduction in bacteria, you have a sexual. Sexual means they can exchange genetic materials, sexual. The first is transduction, is process by which DNA can transfer from one bacterium to other bacteriums by the virus. And it also refers to process ways for reasons DNA is introduced into other cell via viral vectors. Second step, conjugation. In the other slide I just showed you, we have the plasmids forming a bridge between two bacteria, and they can transfer the genes from these plasmids from the first bacteria to second. And the third, transformation. If the negative DNA is taken up from the environment, so for example here some dead bacteria, you have some transfer gene in your environment, and sometimes bacteria will end up this kind of genes called the transformation. Or transformations like we insert the plasmid, the combination DNA, the recombinant DNAs into the virus. So it means that one virus uptakes the change gene from environment. We call the transformation. Next, bacterial culture. How we can culture them? Nuôi cấy culture is here. Không phải văn hóa mà là nuôi cấy. How we culture bacteria? You can culture on agar plates or in liquid mediums. We have a lesson. The last lesson we study about how to culture the cells. You can discover more about how to culture bacteria. But there is a fact that less than 2% of bacteria can be cultured. In the laboratories. It means more like 99% of bacteria and also the archaeas, we cannot culture them, we cannot raise them. And these are in our bodies, like 50% of the our microvirus can culture. And for our body size, this is unknown, but slightly similar to that found in mouse or higher. And total in the world, there's only 200,000 bacteria has been so far cultured. So it means that the knowledge of us about the bacteria is quite limited, because we just can culture just 1% of them. For the others, no information, the way they all these mechanisms happen inside the bacterium knows, very difficult. Once we can culture them, we can perform the research about them. But when we cannot culture, there's very limited information about them. So they are mystery. to the humanity. The virus about prokaryotes, they are decomposure, recycling nutrition from dead to living. Nitrogen fixation, they are only organisms in this planet can fix nitrogen from the atmosphere. They can fix in ammonia. So here's the root of the beans, right? Dễ cây hoạt động các bạn đã có cái kiến thức này từ hồi phổ thông rồi. Dễ cây hoạt động là sự cộng sinh giữa các vi khuẩn cố định nitơ và các cây hoạt động. Vi khuẩn cố định nitơ là only organism in this planet. Sinh vật duy nhất trong trái đất này có thể cố định được nitơ từ trong không khí thành dạng amoni. Và cây sẽ sử dụng amoni của vi khuẩn và cây sẽ cung cấp lại các chất dinh dưỡng cho vi khuẩn. Đây là sự cộng sinh trong rễ cây hoạt động, đúng không ạ? So here's ammonia or other forms of nitrogens are not really remembered, but I remember the ammonia. And we have certain types of bacteria species like, Rhizobium or Azotobacteria. Okay. The next, they can have in the digestion. So we have an abundance of the E. coli in your stomach, in your intestines, actually in your intestines. They can have to digest foods, and also they can have produce some kinds of vitamins that we cannot produce. It is vitamin K and B12. And also, they can produce foods like fermentation food, yogurts, and medicines. Like we can use the bacteria to produce human insulin by recombinations DNAs. And besides the benefits, we have some advantages of the bacteria. They have the pathogens. They are the pathogens. They infect and cause some disease in human and plant and animals or cells. More or less 5% of them are pathogens. And they have a long history in infected and causing the human disease and the death, right? Only 5% of them. So there's so many spoken numbers. We have less than 2% of them. We can culture less than 2% of them. But 5% of them are pathogens cause the death of so many plant, animals, and humans in our history. So how to cause or how to kill them? When you infect by the bacteria, when you have disease by the bacteria, how we can kill them? Go to meet some to see a doctor, and they will give you some drugs and describe drugs. This is antibiotic. Thuốc kháng sinh đúng không ạ? So antibiotics is formed from antiviruses. This means against life. The substances delivered from microorganisms are produced synthetically to kill or suppress the growth of other microorganisms. We call the antibiotic. Simplifying the way we understand antibiotics is to kill bacteria. How this works, or how the first antibiotics prevent? So there is Scottish bacteriologist Alexander Fleming who the first discovered the antibiotic in 1928. He discovered penicillin. he counters the bacteria in the disease. And he goes on vacation. After come back from his vacation, he discovered that some green mold called the Penicillium notatum contaminated, gây nhiễm với các Bacteroides that they grow, his grow bacteria. And it were killing some of the bacteria. And he think that, okay, these kinds of molds can kill bacteria, and we can use that kind of mold to kill bacteria, infect its human bodies. And from that, he can isolate one kind of compound called penicillins, and it's the first antibiotics of the humans. Kháng sinh đầu tiên được phát hiện bởi một sự tình cờ bởi nhà bác học Alexander Fleming. Ông ấy đang nuôi rất nhiều bác đĩa vi khuẩn. Sau đó thì vô tình ông đi nghỉ. Sau kỳ nghỉ về thì ông thấy rất nhiều các loại nấm mốc màu xanh mọc trên các đĩa đó. Và các loại nấm mốc này có thể diệt các loại vi khuẩn gây bệnh mà ông đang nuôi cấy. Và từ đó ông nghĩ rằng những nấm này có thể tiêu diệt được vi khuẩn. Và sau rất nhiều các bước thì ông đã isolate được một chất được gọi là penicillin từ cây nấm Penicillium notatum, và đó là cái thuốc kháng sinh đầu tiên của loài người được phát hiện. So classification of the antibiotics. In the three ways can classify the antibiotics based on mechanism of action, based on the strict spectrum of activities, and based on the mode of action. So first, based on mechanism of action. It can inhibit the cell wall synthesis. You know, our other humans have no cell walls. We can target the cell wall bacteria to invent some kinds of antibiotics. You have penicillins, cephalosporins, or vancomycin. Or second, we can inhibit some function of stellar membranes, polymycins. Inhibit the protein synthesis, we have chloramphenicons, macrolides, clindamycin, tetracyclines. Inhibits the nucleic acid synthesis, we have antibiotics belong to quinolones or ripamycins, or inhibits of folic acid synthesis. Folic acid is quite important to the bacteria. We can inhibit the synthesis of this folic acid. We have the sulfonamide and trimethoprim. And here, based on mechanisms, I have the graph, and it's more easier to follow. Attack the cell wall synthesis, we have beta-lactam, vancomycin, bacitracins. Nucleic acid synthesis, we have sulfonamide, we have the quinolone affect the DNA ligase. We can ripamycin inhibit the DNA polymerase. Protein synthesis, we can attack the 30S by tetracyclines or 50S. Right here, maybe chloramphenicol is more familiar with you. And you have more than 50% of the bacteria antibiotics is focused on protein synthesis. As a lesson about the ribosomes, I have to introduce for you. More than 50% of the antibiotics is target on the ribosomes. Based on the spectrums, we can divide in narrow and broad spectrum. Spectrum of phổ, các loại phổ kháng sinh ạ. Các bạn sẽ nghe hơi quen tai nữa, kháng sinh phổ rộng và phổ hẹp, đúng không ạ? Narrow spectrum is drugs, antibiotics that only act on one kind or one strain of bacteria. But more of them is broad spectrum. Disease can have a wide antimicrobial scope. We have tetracyclines and chloramphenicol is two famous candidates. I think it's when you hear that sound, it might be familiar to you là kháng sinh đúng không ạ? Tetracycline chắc là từ bé mọi cũng đã nghe một vài lần. Tetracyclines is broad spectrum. And these instructions of some antibiotics. Penicillins only kill the gram-positive. Amoxicillins and augmentins. So here I will focus on amoxicillins and augmentins only. I think it's familiar with you when you have a sore throat or you need some kinds of amoxicillins. Sometimes when you cannot get better with amoxicillins, maybe when you the augmentins, right? So the amoxicillin is very common type of antibiotics. And augmentin contains more amoxicillins and clavulanic or clavulanic acid, and it's more effective against some types of infections. So augmentins is fights against broader range of bacteria than amoxicillins alone. So augmentins is we consider the stronger antibiotic than the amoxicillin. Nào về phổ kháng sinh có rất nhiều điều phải nói. Nếu các bạn bị viêm họng thôi đúng không? Các bạn đi uống và cái mà phổ biến nhất là các bạn sẽ có thể uống được amoxicillin. Cái viêm là amoxicillin ở tiếng Anh, amoxicillin quen thuộc không ạ? Từ bé rồi hay là cứ uống thôi không cần biết là mình đang uống cái gì? Amoxicillin. Đến một lúc nào đó amoxicillin mà không có tác dụng lắm, mà thường bây giờ thì có thấy là các bác sĩ hay nhét luôn cho uống augmentin nữa. Augmentin nó chỉ khác amoxicillin, nó chính là amoxicillin cộng với một loại axit là axit clavulanic acid, thì nó mạnh hơn, phổ rộng hơn, giết được nhiều vi khuẩn hơn. Cái điều này các bạn nào cũng có thể chú ý, ví dụ như các bạn vẫn cảm thấy perfect với amoxicillin, bây giờ các bạn uống amoxicillin khi các bạn đau họng nó vẫn có tác dụng tốt, có thể lâu một chút nhưng vẫn tác dụng tốt thì thôi không cần dùng đến augmentin. Điều này xảy ra với thực ra là con trai cô 18 tuổi mới bắt đầu bị viêm họng, đấy là lần đầu tiên cô phải quyết định là dùng kháng sinh cho nó. Thế nhưng mà khi mà đi vào bệnh viện thì bác sĩ đã cho luôn augmentin, một phát cho augmentin luôn, và cô phải hỏi luôn là ê bác sĩ ơi có cái nào nhẹ hơn không? Thực ra cô biết rồi là amoxicillin sẽ nhẹ hơn. Đấy lúc bác sĩ mới bảo là thế uống amo nhé, tôi bảo yes, ok, kêu vào cho em. Thế xong rồi thì cô cũng bảo là thực ra đây là lần đầu tiên cháu uống kháng sinh và em nghĩ không việc gì phải uống đến augmentin cả. Đúng là ban buổi sáng khi đi khám thì ra lúc đấy nó không thể bú, không thể ăn, không thể làm bất cứ điều gì, nó cực kỳ đau, nhưng mà uống amo đến chiều là bắt đầu tươi hơn hớn là đã bắt đầu diệt được vi khuẩn gây viêm họng, đúng không ạ? Đây là điều khi mà các bạn một cái tình trạng mà rất là báo động hiện nay là tình trạng kháng thuốc, đặc biệt ở Việt Nam và các nước kém phát triển, đúng không? Ngay cả người dân thì cũng thích là các thuốc mà chữa bệnh nào nhanh khỏi, đúng không? Bác sĩ thì cũng rất vui vẻ cho các kháng sinh phổ rộng, liều cao, thế hệ mới. Các bạn lưu ý ví dụ như là xem phổ này, cảm thấy nếu mình có thể hạ liều được thì có thể suggest với bác sĩ là mình uống phổ hẹp hơn và thế hệ cũ hơn cũng được. Đối với các bạn khỏe mạnh, bởi vì không phải lúc nào mình cũng yếu, mình cứ dùng kháng sinh triền miên để mình phải tăng liều hay là tăng thế hệ, đúng không ạ? Rồi, so it's about the spectrums of the antibiotic. Next, broad spectrums may use to lead to development of the super infections. When you use the two broad spectrums of antimicrobials, normal microbials, its traits, and the broad spectrum antibiotic will kill the non-resistant cell, the killer cells. And for drug-resistant pathogens, now they have no competitors. They can grow more, and they can cause super infection. So that's why, again, please use the narrow sections if you can, or if it depends on the DT also. But you can use the narrow sections. You can use the narrow sections compared to the broad section. Next, on the basics of the mode of action, you can divide the bacteria into two types. Bactericides. The antibiotics destroy or kill the bacteria. We have examples of Chlorpenicum, tetracyclines. Bacteria's statics is stop or slow the growth of bacteria only. And we have examples, vancomycin, quinolone, rifamycin is very familiar antibiotics, antibiotics for use if you care about the drug you take, okay? Side effects. Some people taking antibiotics will experience common side effects like nausea, diarrhea, stomach problem, vomiting. For the diarrhea is because most antibacterials when you take is a broad spectrum. Instead of kill the pathogens, it will kill other bacteria in your intestines, right? And it's leading to diarrhea. So that's why when you take some antibiotics, the doctor will use in complementary some like you say, intestines, bacteria strains to get you suffer from diarrhea or to help you better after the diarrhea. Side effect của kháng sinh. Cái này thì gặp ở trẻ nhỏ nhiều hơn vì hệ tiêu hóa của nó chưa hoàn chỉnh như người lớn. Khi mà uống kháng sinh thì ngoài diệt các vi khuẩn gây bệnh trong cơ thể thì nó diệt luôn cả các vi khuẩn tốt ở trong hệ tiêu hóa của chúng mình, đúng không ạ? Nó diệt luôn cả vi khuẩn tốt ở trong ruột. Và một cái tình trạng gặp thường xuyên khi người ta uống kháng sinh đó là tiêu chảy. Vì vậy mà khi mà các bạn uống kháng sinh thì thường bác sĩ hay kê kèm với trẻ con là nhiều hơn, hay kê kèm là các men tiêu hóa, đúng không ạ? Để bổ sung lại các lợi khuẩn bị mất sau khi mình uống kháng sinh. So we have a video to summarize about the bacteria. Can you hear this clearly? Or you can see it at homes or okay. Hôm nay khoa tiếng Anh à? Thi và có nhiều cô cho về sớm 10-15 phút, cho nên là cái vấn này các bạn nghe ở nhà nhé. Thì nữa mình sẽ về sớm một chút. Các bạn giải lao 15 phút. Ở lâu anh học không? Học tiếp nha. Học sớm về sớm trường thi. Okay, we continue the lessons and now we study the part 2 about the out here. Remain a little bit about these 3 domains and 6 kingdoms. You can see the domains. The domains out here is more close to eukaryotes compared to the two bacteria. Why they're more close? They're more like similar. They have similarities like normal bacteria, but they have and they also have some differences. That's why we classified in the phylogenetic tree that the archaea is more close to eukaryotes instead of bacteria. So here I have a takers to compare bacteria, archaeas, and eukaryotes. About the cell walls, peptidoglycan in the cell walls, all bacteria have peptidoglycans. However, even the gram-positive or gram-negative, they do have a peptidoglycans. Archaea, just some, but eukaryotic cells, none of them have the peptidoglycans. Second, about the ribosomes, bacteria 8 prokaryotic 70s, archaeas in between, sometimes 7, sometimes 80, eukaryotic cells 80s. First, amyloids in protein synthesized the methionines and the derivative of the methionines. Archaeas is a methionine derivative, but archaeas is methionine, and the first amino acid is methionines. Our fourth characteristics: sensitivity to streptomycin and prolamphenicones. Yes, bacteria is sensitive to these. Archaeas no, and eukaryotics no. Next, introns in the genes. So you know that in the genes, bacteria very rare are no introns. Archaeas, some genes we discover the introns, but in archaeas they present in so many genes. What are the introns? So here's the genes in the eukaryotic cells. They have some fragment of DNA that containing the information, genetic information. We call the exon. And they also insert some part that is no information. We call the introns. And then the exons, introns, axon. This is in eukaryotes and some genes of archaeas. But in bacteria, very rare or no introns. So for when they insert some part that is no information between the exons, it will increase the stability of the cells. Is less mutation will occur, right? Because there's some mutation occurring in introns, nothing happens. It don't affect the production of the genes. Next, histone presence. No histone present in bacteria, but some are, histone is present in the genetic materials of archaea and also the eukaryotes. Next is RNA polymerase, have one kind, several kinds discovering archaeas and also eukaryotics. Carbon linkage of lipids. This is only thing distinct with the archaea. They are quite special in archaeas compared to bacteria and eukaryote. Because the carbon linkage of lipids of archaeas is ethers, whereas in bacteria and eukaryotes they are the ester, linked ester. And for the rest, we have the ether. Somehow, how this can difference. More specific of the archaeas here. Or just here's in the first part of the pictures, you can see the phospholipids formed in archaea. Here is the linkage. The linkage between the phospholipids here is even ether. Whereas for bacteria and eukaryotes, we have the ester linkage. But the first difference from them, and second difference, in archaeas, the lipid tails is quite different here. They have so many side chains. Too much of the side chains may prevent the archaea membrane from leaking at high temperature. So that's why archaeans can live in very harsh environments, like in very hot environments, right? Because here in the lipids of them, they have so many side chains. And second, in some archaeans, the lipid bilayers is replaced by monolayer. So in mammals, in bacteria and eukaryotic cells, the plasma membranes is double layers. However, in archaeans, their tails will fuse together, so they make only monolayers with two polar heads and only one tail stick, or they fusion the tails together, and it might make the membrane is more rigid. So it's better to resist the harsh environment. So that's why this is very special about the archaeans. So that's why we call them the extremophile. They can live in very harsh environments, too acidic, too base, too toxic tolerance, or too hot or too cold environments. Ok, cái gì quan trọng là tiếng Việt? Ở đây có một cái đặc điểm mà archaea con vi khuẩn cổ nó khác biệt và khác biệt hoàn toàn so với các loại vi khuẩn hoặc là các tế bào nhân thực bình thường, đúng không ạ? Có hai điều. Thứ nhất là liên kết giữa carbon linkage và giữa carbon và lipid của nó là ether, còn bọn còn lại là liên kết ete. Cái thứ hai là side chain của bọn archaea này, có rất nhiều các side chain ở trong cái đuôi của cái phospholipid của nó. Để làm gì ạ? Nó là tăng độ vững chãi cho màng tế bào thôi, chẳng có gì cả. Cái thứ ba, một số loài archaea và thực ra là nhiều con archaea thì nó thay vì có double layer, có hai layer của phospholipid ở trên màng, thì nó fuse, gọi là cái gì nhỉ? Nó tiếp hợp hay là nó link hai cái đuôi này lại với nhau, chỉ thành một đuôi ở giữa thôi và nó sẽ thành monolayer. Và cái monolayer này cũng làm cho cái màng tế bào trở nên vững chãi hơn và có thể chống chịu với tất cả các môi trường rất khắc nghiệt. Vì thế mà mình gọi con archaea là con loài extremophiles, là loài chuyên sống ở những cái môi trường khắc nghiệt mà chả con nào thèm sống nữa. Quá axit, quá bazơ, quá độc hoặc là quá nóng, quá lạnh, toàn con archaea để sống được. Next is about videos about the archaeas. I don't know if there is problem with the sound or not. Okay, I will skip this step and come back if we can fix the problem with the audio. The next part of the lesson today is about the viruses. Viruses are the histology of the virology, the study about the virus, and the infection of the virus begins in the closing years of the 19th century. And it was discovered by Dmitry Ivanovsky about the tuberculous virus. So they are, at the time there's a disease in the plant, in the tubercles, and First invented of them is the scientists bring the leaf of the disease on tomatoes, and then they can spread that kinds of solutions into the and after he filters from the bacteria-free filters. This means there's no bacteria in that solution, and then he spreads into the strong leaf or normal tomato leaf, and then the normal tomatoes will become infected and will have that kind of disease. And they have a conclusion that there's some species or something is smaller than the bacteria can affect or can cause that kind of disease. That is the first time they discovered this tomato mosaic virus, TMV. And these discoveries we consider the beginnings of the virologies. That means viral power. So what are the viruses? So the virus, parasites, it means that they can depend entirely on the other hosts or another living organisms. They cannot be observed under the light microscope, and they have no internal cell structures. They contain either DNA or RNA, but never contain both of them. The tension about that is may appear in the final exams so many times. I think each year, in any years, have this kind of question. They are incapable of replications unless they occupy an appropriate living host cells, and they have no powers of metabolism. And individually, they have no increase in the size. So that some compound in the world have roughly 10 powers 31 viruses of any given moment. So if you can line up them end to end, and the columns of the virus may extend nearly 200 light years into space. So it's a very huge number of viruses in this planet, right? Ở trên hành tinh này có khoảng 10 mũ 31 virus, các loại virus. Và nếu mà mình nối nó lại thành cái cột thì nó dài đến tận 200 năm ánh sáng. And this is the type of sign of the virus. You can see here is the eukaryotic cell with the big nucleus. Prokaryotic cells is more like an organelles of the eukaryotic cells. Like here we have a mitochondria. But here the sign of the virus is like one big molecule only, macromolecule only. There's some kinds of virus. We have the HIV, hepatitis B, Ebola, bacteriophage, and this infectious, the bacteria, rabies virus, virusia, influenza, and so many kinds of the other virus. What is the structures of the virus? It is very simple. For the naked virus, they have just a protein coat we call the capsid, and in the center we have DNA, pay attention, or RNA, never both. And the second type of virus is envelope virus. They have the spines, proteins surrounding the naked virus. And here is, so remember coronavirus? They have the envelope virus. They have so many spikes, proteins surrounding them, and we trigger it, we target on the spike proteins to produce a vaccine to prevent the coronavirus. A virus recognizes cells it can affect by matching surface or markers with the receptor sites on the cells, and certain viruses can only attack certain type cells, and they are said to be very specific. For example, the virus that infect the plants cannot cause disease on humans. And somehow, in generic case, so that for this coronavirus is really that originated from the bat, right? So in our case, the virus that infects the bats cannot cause disease in humans. Then it transforms and in some ways to infect the humans. And the plants, the virus infect the plants, no way to infect, to cause the disease in humans. Here, the ligand receptor is mapped to the surface of the cells. So replication. There is two types of replication of the virus: lytic pathway switching, Xingdan, and lysogenic pathway switching, Diamdan. Okay, so in lytic pathways, DNAs of the virus affect the host cell. They replicate, they synthesize the protein code, and then they lysate the host cells, and really new virus. And the lysogenic pathway, just in Dianban, they just integrate the DNAs into the host cell, and then multiply so many times, inductions. And if there is some stress or when you have the weak immune disease, it can reverse, come back into the lytic pathways and lysate the host cells. So the virus infect the particle that can reproduce or commanding a host cell and use the machineries of the host cell to make more virus. This is lytic cell that the phagostigmate. This is one kind of the virus that affect the bacteria. So whenever you see the figure here, this means virus infects bacteria. So here's T4 bacteria, T4 figures. One, they attack the host cell and they attack the bacteria. Second, they inject the DNAs of them into the bacteria. Next, penetration. The only protein, the fibers inside is only protein synthesis. The host cell DNA will be degraded. Next, replication. DNAs of the virus is multiplied so many times. Next, synthesis of the late structure of the proteins. They use the machine. They use the source, the materials inside the host cell to produce so many other parts of them. And next, they will assemble to be a new virus. Final step, they can really lysate the host cell will die because it is like this. And really, the new viruses, so many new viruses to attack other cells. Here, bacterial fibers. The viruses affect the bacteria. For the viruses with the genetic material RNAs, we call them the retrovirus. Các virus ở phiên mã ngược. Because they are RNAs, when they fuse and they inject these RNAs inside the host cell, for example, the HIV virus, they need a reverse transcription, reverse RNA to cDNAs. cDNAs then integrates into the DNAs of our bodies. And the next, they synthesize mRNA to synthesize the protein of the virus, and RNAs also, the buddings, maturation become the new HIV virus. Rồi, virus HIV là một loại virus mà tất cả lưu ý từ retrovirus, tất cả các virus mà chứa thông tin di truyền là RNA thay vì DNA mình gọi là retrovirus. Thì khi mà nó xâm nhập vào trong cơ thể, trong tế bào vật chủ, thì nó phải có một bước là phiên mã ngược từ RNA thành DNA, sau đó là mới insert cái DNA đó vào trong DNA của vật chủ được. Và nó lợi dụng cái hệ thống của DNA của vật chủ để sản xuất ra mRNA và translate ra các loại protein của nó. Ngoài ra thì nó cũng transcript ra RNA của nó luôn. Và sau đó thì virus thì đơn giản là cấu tạo từ protein và DNA hoặc RNA thôi. Nó sẽ lấy DNA do mình tổng hợp ra, wrap around, bao lấy RNA của nó, nảy trồi và sinh ra rất nhiều virus HIV mới. Đấy là cách nhân lên của virus HIV. Nó ngược một chút, nó thêm cái bước là reverse transcription. The next one type, prions. We believe that the virus is equal to DNA or RNA, both are covered of proteins, right? But here is one thing that still causes the disease. It's not really the virus. We call them the prions. Prions is a proteinaceous infectious particles and a cell replicating proteins responsive for a range of neurodegenerative disorder in human and mammal. And these include the bovine spongiform encephalopathies, or we call the BSE, mixed cow disease, bệnh bò điên, caused by the prions. It's not a virus. Bò điên is familiar with you? And they cause the disease in human cells. Prions, they affect the brain and it's always better. Lúc nào cũng là dẫn đến chết, không chữa được. Series are how could something that contains no nucleic acids be capable of replicating itself? So there are prions. It's just a proteinaceous infectious particle, một phức hợp dầu protein thôi, đúng không ạ? So how could something, they have no nucleic acids, but they have a capable of replicating itself. How? You think it's strange? Tới tiếp đi đúng không?
that causes the mad cow disease is alpha, and the beta type is the one that causes mad cow disease. So somehow the beta type makes the alpha type become the beta. So it's just like protein making itself like more like the one that causes the disease, but not actually the protein synthesis.
I think it's nearly the answers. Okay, it means that prions maybe enter the version of the normal proteins, and somehow they have the ability to convert the normal versions and reform itself into the mutant form. And that's why this is the way the prion can propagate itself, right? So it's make the normal proteins refers itself to become the mutant one. This is how the prions can replicate these cells. Second type, viruses. So the virus is a plant pathogen that comprises only single chain RNAs and do not code for any proteins, but it causes disease also. So prions is just proteins cause disease, and virus is RNA or DNA cause disease without any protein code outside. This is one aspect of the virus. Here is so whenever you see ssRNAs, it means that single strand. And when you see dsRNAs, it means double strand. Now, here's my opinion final exams. Ok, đây có dẫn gì? Trong đề thi cuối kỳ thi thoảng các bạn sẽ nhìn thấy là ssRNA, ssDNA hoặc là dsRNA, DNAs. SS thì nó stand for single strand, sợi đơn ạ. DS là double strand, sợi kép hoặc là sợi đôi ạ. So in 1971, Dyers, they proposed the name virus to describe the new discovered pathogen of the potatoes. The virus, many times smaller than the small leaf virus, they consist only small speckles of the single-strand RNA, just 30 to 400 nucleotide base, no protein code. And they cause pathogens in potatoes, they just only single-strand RNA. In the previous part, only proteins cause disease. And next part, we have some comparison between the virus and the normal cells. The virus structures of only RNAs or DNA cores, and the protein core we call the capsid. By the cell, we have a servers of basic components, cell membrane, cytoplasms, genetic materials, DNAs, and organelles. We have a ribosome in prokaryotes and so many high complex organelles in eukaryotic cells. About replication or reproduction, is copy itself only inside the host cells. Without the host cell, they cannot multiply. So, for example, the coronavirus, when you spread the coronavirus by couchings, they just stay there in the environment, dying or stay there, no replicate. It only replicates inside our bodies, inside our cells. For the cells, is can replicate sexual or asexual. This means there is genetics transfers or no genetic transfer. Next, about the genetic materials. In the virus is DNA or RNA inside of the virus. But for the cell, they have a both. They have a DNA in the center and have an RNA also. For the growth and development of the virus, there is no increase in the size, no decrease in the size. There is no change in the development, in the shape of the virus. But for the cell, yes, in the multicellular organism, they can grow, they can develop. Even in the unicellular organism, you can see the size increase a little bit after they divide. Obtain and use the energies, no for the virus, yes for the other cells. Respond to the environment, no for the virus. Yes, for the cells. For the virus, if environment is not suitable, they just die. Actually, there's no response from them. Dies only, just walk. Change over time, no for the virus, but yes for the cells. Skip this. The virus are living things. You think that the virus are living thing or not? Half, yes. For scientists, it's called the virus is tides of living. In Vietnamese we call it nó là một dạng sống. We do not call them, and instead for the rest we have a sinh vật sống, right? Or sometimes you read in the news, even in the VTV3 or in the videos, or sometimes they have a misunderstanding of the virus, they call the virus is sinh vật or something like this. It's not true. The virus is just dạng sống, they are not sinh vật. So the function of the virus. The important virus, most virus is the virus is harmful. It's called the disease. We call it pathogenic. The disease producing agent we call the pathogens. There is a lot of human disease as caused by the virus. We have the worst common colds, influenza, smallpox, Ebola, hepatitis, AIDS, chickenpox, rabies, so many diseases caused by the virus. Here is smallpox lesson. Bệnh gì đây ạ? Đậu mùa đúng không ạ? Smallpox. The virus will disrupt the body's normal equivalent or disrupt the body's balance, and it can be prevented with the vaccines, but not treated with antibiotics. Antibiotic is just treat the bacteria. For some house, for example, when you have been infected by the coronavirus, somehow you can use the antibiotic, but it's not for the treatment of the coronavirus. It's just when you get the coronavirus, your bodies will be weaker, immune system is not strong like normal, and some other bacteria can invade and causing the disease, and the antibiotic will be help to destroy other bacteria, but not for the coronavirus. And also, we have the beneficial from the virus, like genetic engineering. The Hamlet virus can carry good gene into cells. And in this, do you know this, or or we can use the virus to produce the vaccine, also the Hamlet virus to produce the vaccines. So guys, for ICT, I want to ask a question. Can you tell me the common between the virus in the IT fields and in the biology field? Do you see any comments? So that's why this creature in biology, they call the virus also. I think the virus of the ICT is newer compared to the virus in biology, right? Maybe the virus in ICTs is originated from the virus in biology. What is common between them? Like the virus can affect our laptop and the virus affects our bodies. What is common between them? They are the commanders, right? They command the host cells and use the resource of the host cell to make small virus. You think that's the same between the viruses of the computers and the virus affect your bodies? Or somehow they can basically reprogramming to become a virus factories? Right? So the virus is enemies of these computers, and this is the enemies of the body of the cells. We have videos about the virus, but maybe we'll see it in homes. Over here is a small part about the coronavirus. And this is how we can produce the Pfizer. How we can produce the Pfizer vaccine of the coronavirus. Formulation with the nanocarriers. We have the mRNAs. We have the lipase. Lipase in the water will automatically form a liposome covering the mRNAs. But once special years, NLPs, this is not nanolipids. This is liponanoparticles, nanoparticles. It's for nanos, student from nanos, it's made from use. You invent for this. And so many ways of deliveries, the drug, liponanoparticle formation. They can form to make liposomes. We put in a saline solution, inject in the humans, and then this small liposome will be under cytosis into our cells. Relieve the mRNAs. mRNAs will be synthesize the viral proteins. This is the spike proteins of the coat of the coronavirus. And then ours blood sound with digest them, sending to MHC1, MHC class 1, and then send information to the T cell by no distant communication, right? Remember this? Okay, it's all about these virus. Yes. We have another videos, but maybe you see it at home because the problem with the audio system in this auditorium today. I don't know if this cannot. We cannot hear the audio system of these auditoriums. So thank you very much. The lesson will end today. We close 15 minutes sooner for you to prepare for the test this afternoon.
저희가